I'll be talking about B cells, which actually work closely with the TFH and TFR cells that Mizuba mentioned. And specifically, I'll be talking about developing and harnessing a technique to study HIV-specific memory B cells. So a memory B cell is a cell that recognizes familiar pathogens that the body has been exposed to before. A memory B cell is coded in antibodies that bind specifically to a pathogen that the body has been exposed to before. And so it's been shown that HIV infection is associated with poor B cell responses. In addition to that, memory B cells in acute HIV infection remains understudied. And so the goal for my project was to develop a technique to be able to study these HIV-specific B cells in patients with acute HIV infection. And so in order to develop a technique, I focused specifically on a method called flow cytometry, which essentially allows us to study specific cells and cell populations within a larger population. And the way that it does this is by using probes and fluorochromes. So a probe essentially just binds specifically to our cells or population of interest, and a fluorochrome, which is attached to that probe, essentially just releases a wavelength of light that is very specific that the flow cytometer is able to detect, and from there tell us what we're looking at. And so in order to produce a probe that binds specifically to these HIV-specific B cells, I essentially conjugated two different things. The first was a tetramer fluorochrome, or rather a tetramer binded to a fluorochrome. So tetramer is just a protein with 4 different subunits to it. And again, our fluorochrome is just our light-emitting particle. And conjugated to this was something called our SOCIP. So SOCIP is basically just a mimic of an HIV envelope. So as I mentioned, memory B cells are very specific. And so HIV-specific memory B cells bind to HIV. And so it's important that our probe had this SOCIP because that was how specific HIV B cells would be able to bind to the probe. And so as we see in our completed probe, we have the fluorochrome attached to 4 of the SOCIP molecules. And so the reason why we have 4 in this tetramer apparatus is so that we can increase the specificity of this binding. And so from here, we were able to put the probes in, for example, a bulk sample of cells, and the HIV-specific memory B cells would bind onto the probe. And from there, we can quantify and analyze using flow cytometry. And interestingly, we did this not with 1 probe, but with 2 probes. So we made 2 different probes, and the difference between the probe was the fluorochrome. So each probe had a different fluorochrome that emitted a different wavelength of light. And the reason that we wanted to do this is because we wanted to be able to identify cells that binded to each probe. The reason we wanted to do this is because we wanted to be specific. We wanted to know that these cells were indeed HIV-specific B cells. And indeed, this double positive probe staining approach proved to be thorough. On the left, you'll see a sample from an HIV-negative patient. And within the gate, within the square, we see that 0% of the cells binded, and that represents no cells binding to both probes. And if we look at the chronic HIV patient, who we would expect to have HIV-specific memory B cells, we did see a good amount of binding to both probes. And so we see that this double positive approach is effective and that it works for identifying HIV-specific B cells, since it worked in an HIV patient, but it didn't work in an HIV-negative patient. And so from here, we use this approach to generate some preliminary data. Again, we're addressing this initial problem of trying to understand more about HIV-specific B cells in patients with acute HIV infection that were treated early. And so the first thing that we looked at was looking at the percentage of HIV-specific B cells out of total B cells in acute HIV patients treated early. And what we found was that the percentage of HIV-specific B cells decreased in patients who were treated early. And to us, this made sense because if you are treating a patient with, for example, antiretroviral therapy, they're going to have fewer copies of the virus within their system. And because of that, they'll need fewer B cells to be able to fight the infection. And so this made sense. And so next, we use this approach of studying HIV-specific B cells to characterize these B cells into one of 4 different categories. So memory B cells come in all different shapes and forms. There are 4 to be specific, and we use flow cytometry and additional probes to take this subset of HIV-specific B cells and learn more about them. So we're able to use these probes, these additional probes, to basically characterize them into one of 4 subset. And this again just underscores the power that this method has to learn more about these HIV-specific B cells. And so to conclude, this method was effective. So I was able to develop a robust probe-based technique to study HIV-specific B cells. And we see that through this preliminary data that this indeed is an informative way to learn more about these B cells. And so in the future, we plan on studying more samples. We want to know what happens at additional time points. We want to know what happens at pre-infection. And from there, after identifying these specific memory B cells, we want to perform additional characterization. So in addition to learning about what subset these B cells belong to, we want to learn stuff like how proliferative are these cells and other characteristics. And from there, this has the potential to inform us on vaccine and cure studies because we'll be able to learn more about the effect of early treatment on HIV. And so as you've seen, us labbies, as we call ourselves, we're able to study all different types of cells in relation to the HIV infection. And we learned so much about these cells, but also about HIV and about the interesting field of immunology. And these are definitely lessons that we'll be taking with us back home. And these are lessons that we couldn't have learned without our incredible mentors and our PI, Zaza.